in these breaks, there is the possibility, if you wish, to listen to a 15-minute presentation from partners, so-called partner talks. And the first one today will be from, in this break, will be Kirsty Mchugh from Oxford, talking about antibodies to malaria. So I'm not sure about the best format to do this. I guess if folks plan to leave immediately, they probably should do so. And those that want to stay and listen to the 15-minute talk, stay. Okay? Well done.
Yeah. Yeah, it's fine. These lights going to be turned down. Are these lights going to be? Okay, so I think I'll begin. First, I'd like to say thank you very much to the organisers and to the Cartera team for the opportunity to present here today. And also thank you for those who have stayed. I hope that it's worth it for you and you're not missing out too much on the coffee. So my talk today is on the high-throughput isolation and characterisation of monoclonal antibodies to Rh5, which is currently the leading blood-stage malaria vaccine candidate. So what I'd like to do is just to start with a quick overview of our research group and then introduce blood-stage malaria and the Rh5 antigen before spending the remainder of the talk focusing on our pipeline for isolating and characterizing antibodies and how we've used this pipeline to generate a panel of Rh5-specific antibodies from our VAC63 clinical trial. And I'd also like to highlight how the Cartera LSA platform fits into our workflow. So I'm a senior postdoc in Simon Draper's Blood Stage Malaria Group in Oxford, and the focus of our research is very much on vaccine development and antibody immunology, mainly relating to malaria. And really one of the key strengths of our group is that we're able to carry out preclinical vaccine development in parallel with our own early phase clinical trials. So we have a very active clinical team who've undertaken 16 early phase clinical trials to date. There are 4 currently in progress, and they've also developed controlled human malaria infection models for the 2 major species of Plasmodium that cause human malaria. And in terms of our lab research interests, they cover 3 main sort of broad areas, but what I'll focus on today is our antibody work. And here, what we're looking to do is to generate large panels of antibodies from our vaccinees and use these antibodies to generate epitope mapping information and functional data of these antibodies. And if we can combine this, it will allow us to better understand the vaccine-induced antibody responses. And then we can use this information to inform next-generation vaccine design and also hopefully to develop the first blood-stage malaria antibody therapeutics. So for those of you less familiar with malaria, it's caused by the single-celled Plasmodium parasite. And so very briefly, the infection begins with the bite of an infected mosquito. This deposits a form of the parasite into the skin called the sporozoites. These sporozoites rapidly migrate to the liver and infect hepatocytes, and then spend the next 7 days developing into the next stage of the parasite, which are the merozoites. These merozoites then rupture out and into the blood, and here they can infect red blood cells, and this is the beginning of the continued reinfection and exponential growth phase. And it's this blood stage that's the cause of the clinical symptoms of malaria and is responsible for the associated mortality and morbidity if the infection isn't brought under control by the immune system or by anti-malarial drugs. And then just to quickly finish the cycle, some of these asexual stages go on to form gametocytes. These gametocytes get taken back up into the mosquito, they fuse, and then undergo a complicated process to form the sporozoites again. So this complex life cycle offers several opportunities for immune intervention, but as I've mentioned, we're focused on the blood stage, but there have been a number of problems or difficulties facing us when developing a blood stage vaccine, and this is mainly related to the significant antigen polymorphism, the use of redundant invasion pathways, and the need for really high concentrations of antibodies. So when it comes to RH5, the discovery of the RH5 antigen really represented a breakthrough in the field because this was the first highly conserved merozoite target that was identified and susceptible to vaccine-induced antibodies. So RH5 forms an essential interaction with basogen on the red blood cell surface, and this is shown on the left where you can see the structure of RH5, which is formed of these 2 3-helical bundles that come together to form a diamond shape. So that's shown in blue in the top left, and it's at the tip of this diamond that's the interaction site for basogen. And we now know that RH5 is actually part of a much larger invasion complex, this so-called PCRCR invasion complex, which is made of 5 essential conserved proteins. So this also includes pTRAMP, CSS, RIPA, and CYRPA. So these additional antigens are now also under active research in our group as potential vaccine candidates. But going back to RH5, so how has it performed in the clinic? So 4 early phase clinical trials of RH5-based vaccines have now been completed. RH5 has been delivered using different platforms and formulations in both the UK and in Tanzania. And I'm not going to go through this clinical data today, but just to say that in the UK trials, the VAX63 RH5 protein in adjuvant vaccine gave improved antibody quantity and functional activity compared with the VAX57 viral vectors. So for the purposes of the antibody work that we've been doing, it was the VAX63 volunteers that were selected for MAB isolation to map the RH5 epitope landscape. So how do we do our antibody discovery? This slide, this is an overview of our pipeline, and I'm sure it's probably familiar to many of you in similar formats. But what we do is we start with PBMCs from our vaccinated volunteers, and then with or without a B-cell enrichment step, we stain these cells with antigen probes and phenotypic markers for single-cell sorting, and then this is followed by RT-PCR and then sequencing, and then the paired heavy and light chains that we get back from sequencing are sent to Twist Bioscience, who synthesize these inserts and clone them into our custom vectors. We then use these vectors for small-scale transfection of hex cells, and then we purify the supernatants using protein A chromatography on an ActaPure system that we have linked up to a Teledyne autosampler. So this allows us to run up to 84 antibody samples in a single run. So this whole process takes about 5 to 6 weeks, and it gives us about 0.5 mg of antibody, which is enough to do our downstream initial characterization. So from the VAX63 clinical trial, we took 21 volunteers and isolated 240 antibodies. So these antibodies have been characterized in a number of ways, and here we're really looking at the potency, whether there is any synergy or antagonistic relationships between these antibodies. But really, one of the limitations that we were finding is when you're working with such large antibody panels, it's very difficult to get full kinetics and epitope binning information. So to address this bottleneck, at the end of last year, we purchased the Cartera LSA platform. So that's shown here. And so this platform has a number of benefits. The Cartera team are here today, so if you want to discuss any of these benefits with them afterwards, you're more than welcome to join them at their stall. And I should also point out that Fair Journey Biologics also offer this Cartera LSA service as a service. So you can come to Fair Journey Biologics to use the LSA. But what we use the LSA for is to do the epitope binning of all our antibody panels, high throughput kinetics, and we've also started to carry out peptide epitope mapping as well. So for those of you that don't know about the LSA machine, I just want to give a very quick overview. So it uses traditional SPR with a gold surface, but this is integrated with advanced fluidics. So it has two channel modes, a multi-channel mode and a single-channel mode. So on the left, if we look at the multi-channel mode, there are 96 needles that are lowered into a ligand plate to take up the samples. This printhead then docks onto the gold surface to either capture or couple the array using a bidirectional flow. So if you could play the video of the left. So this shows this process happening. And this can be repeated up to 4 times to create a 384 array, which can be either of unique ligands, replicates of your ligands, or a concentration series. And then once your array is generated, the single flow cell can then be used. So this docks onto the chip to deliver either buffer blanks, your antigen, or any regeneration buffer. And this is over the entire ligand array in single injections. So if you could play the video on the right, as demonstrated here. So moving on to our data, so to do our epitope binning of our large antibody panel, we took our 240 antibodies, but also combined it with 8 sentinel antibodies that had been previously characterized from a smaller study. So these antibodies were coupled to the chip to create the ligand array, and then our RH5 analyte was injected over the whole array with the single flow cell. And then the first of the coupled antibodies was then injected as an analyte. And then following binding, the ligand array gets regenerated for the subsequent cycles with the remaining panel. The data can then be processed using the epitope software, and this generates a heat map, which is shown on the right here. So you can see the antibodies that compete with each other are shown in red, whereas those antibodies that don't compete and are able to sandwich are shown in blue. So this data can also be represented as a dendrogram or a network plot. And what we've done here is we've used our sentinel antibodies to benchmark the epitope binning assignments. And this has allowed us to group our antibodies into 10 high-resolution epitope communities, which are shown in both diagrams. And really what the power of using this epitope binning on such large panels is it allows you to pick up the very rare antibodies that perhaps display more nuanced blocking behaviors, such as these antibodies in 4C that show crosstalk between 2 of the epitope communities. And these might be antibodies that are of particular interest. So what you can also do is we can combine this epitope binning data with some of the structural information we have from our sentinel antibodies. So in the top left, in gray, you have the RH5 diamond structure, and there are 4 structures of antibodies from our sentinel panel, shown in red, blue, green, and orange. And they're also labeled on the bottom left network plot. So all of these antibodies had come from different communities. And then what this really allows you to do is look for the potential binding site regions for the remaining communities. And you can predict these based on the overlapping competition profiles. So for example, if you take that yellow epitope community 5B, that is situated between the red and the orange bins, and so we can sort of estimate that the epitope lies between that on the structure. But of course, this isn't a replacement for high-resolution structural data. However, what it does do is it allows you to, it informs the down selection of antibodies for structural studies so that you can precisely define any missing epitopes. So that's what we've done. We've now used this data to select 10 further antibodies to take through to crystal studies. So you can also combine your epitope data with functional activity. So I've shown on this slide 2 different examples of this. On the left, we've overlaid the protein blocking activity of our antibodies as measured by BLI onto the network plot. So in red, you can see all of the antibodies that are able to inhibit basogen binding, whereas in blue are all the antibodies that can block CYRPA binding. And that CYRPA is that one of those proteins that's part of the big invasion complex. And then on the right-hand side, we've got another example. So this is where we've overlaid our parasite growth inhibition activity of the antibodies onto the network plot. And here, the antibodies that are labeled in red are the ones that have high growth inhibition activity. So these ones are inhibiting, these are our most potent inhibitors. And then if you compare the two data sets, you can actually see that all of the antibodies that block basogen have high GIA activity, whereas all the antibodies that are blocking CYRPA in blue on the left have very little or no GIA activity. And then the last thing we can do is we can also use, we can combine our epitope data with sequence information. So here we've plotted so here we've shown all the unique combinations of the germlines that we have in our panel. But on the bottom, we've plotted the growth inhibition activity against the most common gene combinations in our panel. And then we've colored these according to the epitope community. And actually, this reveals a subset of antibodies that are very potent, all share the same gene combination, and are all part of the same epitope community red, which is shown on the bottom right. This data should all be disclosed on bioRxiv as a paper in the next few weeks, so the potent gene combination will be revealed then. And so I am running out of time, but I just wanted to quickly show some of the kinetic data that we have. So I have the experiment overview shown on the left, but essentially here we can capture our antibodies on an FC lawn in the multi-channel mode, and then flow our analyte over the top in a threefold dilution series. And then following each set of analyte injections from the lowest to the highest, this ligand array then gets recaptured to allow repeat experiments to be carried out. The kinetic parameters then can be calculated using the Cartera Kinetic software package. And on the right, I've just shown one of the sort of useful features of the software package is that it automatically flags ligands for further investigation, depending on the criteria that you set. So here I've shown in yellow the flags show where you've got residuals more than 7% Rmax. In grey, these are the antibodies that have quite low RU values, so they're likely to be non-binders or very weak binders. And then in pink, we've got an antibody that has very, very slow dissociation. So I'm not going to show all of the data, but here is an example of some of the sensograms from some of our panel. And we've excluded those that were grey flagged, but you can see here that we've got a number of antibodies that have very long dissociations, and just a couple that in yellow that have been flagged that didn't quite, that had higher residuals. So this data can be plotted on an isoaffinity plot, which is shown in the top left. And again, we can overlay our epitope binning data on this to look to see if there are any relationships between the binning and the affinities. And what we have is we have a nice range of affinities from about 30 picomolar up to 30 nanomolar. So we do have some very, very high affinity antibodies in our panel. But there doesn't seem to be any association with epitope community, because in the bottom right you can see that there's a quite large spread of the affinity constants across the whole of all of the communities. So I'm going to stop there. I do have peptide mapping data, so if anyone is interested in the peptide mapping application using the LSA, then do please come and talk to me, and I'm happy to talk about our experience with this feature. But I'll just quickly skip to my summary. So we've established a pipeline for our antibody isolation and characterization that's really set a paradigm for us for our future antibody campaigns. We've characterized the epitopes on RH5 to a very detailed resolution. And really, by combining the epitope information with function and kinetics and sequencing information, we've revealed some determinants of potency that are really going to help inform our next generation vaccine design. And while I haven't shown this today, we have identified some very potent antibody candidates that we're taking through for therapeutic development. And we've also started to look at combinations of these, and it's looking quite exciting, and we're revealing some new synergistic relationships between our antibodies that will also inform our next generation vaccine design and combination therapies. So that's just to thank everybody in Simon's group and all of our collaborators. Thanks.